Assertion reasoning. During respiration, the ultimate fate of pyruvic acid depends on the availability of O2. When O2 is available, it is aerobic respiration. Pyruvic acid moves from mitochondrial, what you call cytosol, to mitochondrial with the help of a special protein carrier called pyruvate translocator. So when oxygen is not there, it remains in the cytosol and undergo fermentation to produce ethyl alcohol. So reason, the special transport protein pyruvate translocator transports pyruvic acid from cytosol to mitochondrial matrix. Second is correct. That means reason is correct, assertion is correct, but reason is not the correct explanation of assertion. So A and R are correct. R is not the correct explanation of A, second option. Complex 1 of electron transport system of mitochondria is considered as gateway of electron transport. So complex 1 of electron transport system of mitochondria is considered as gateway of electron transport is correct. Complex 1 contain FAD as its coenzyme is wrong. Complex 1 will be like this. It extends from intermitochondrial space to mitochondrial matrix through inner mitochondrial membrane. Here it has FMN, flavin mononucleotide, as prosthetic group, and more than or many iron sulfur proteins. It takes electrons from NADH2 and transfers here, and protons from mitochondrial matrix and transfers in intermitochondrial space. So FMN is the prosthetic group of complex 1 NADH dehydrogenase complex. So complex 1 of electron transport system of mitochondria is considered as gateway for electron transport is correct. Complex 1 contain FAD as its coenzyme is wrong. Complex 1 contain FMN as its coenzyme is correct. So A is true, R is false. ATP synthesis in mitochondria is closely associated with. ATP synthesis in mitochondria is nothing but electron transport system where reduced coenzymes involved in oxidation and ATP formation occurs. It is not release of CO2, it is not reduction of NADP and FAD, it is not release of oxygen, it is the consumption of oxygen. Why? 2 electrons liberated from complex 4 ultimately 2 electrons liberated from complex 4 ultimately goes and attaches to half O2 where 2 protons are added and water is formed. So ATP synthesis in mitochondria is closely associated always with consumption of CO2. That's why that process is also called oxidative phosphorylation. That means ATP formation in the presence of oxygen, or during oxidation ATP is formed. So ATP synthesis in mitochondria is closely associated with oxidation, that is consumption of oxygen. Choose the correct ascending order of the following with reference to the number of ATP formed in aerobic respiration. 1 molecule of acetyl coenzyme A 14, 1 molecule of pyruvic acid 15, 1 molecule of glucose through ATS 36 or 32, 1 molecule of glucose in cytosol only 4. So answer is D 4, then A acetyl coenzyme A 13, then B 15 1 molecule of pyruvic acid, so acetyl coenzyme is 12, 1 molecule of pyruvic acid 15 and 1 molecule of glucose through ETS 32. So D A C B, 1 molecule of glucose in cytosol only 2 ATP, 1 molecule of acetyl coenzyme A. So first D, next A, then B, finally C, because 32 ATP are formed when all the reduced coenzymes involved in electron transport system. This is an assertion reasoning. Glycolysis is a prerequisite for fermentation. Glycolysis is a prerequisite for fermentation. Whenever glycolysis takes place, glucose by glycolysis in cytosol produces 2 pyruvic acids, 2 NADH2, and net gain is 2 ATP. If this pyruvic acid remains in cytosol, it involves in fermentation. Suppose this pyruvic acid goes to mitochondrial matrix with the help of pyruvate translocator, the protein carrier called pyruvate translocator. It involves in Krebs cycle. It involves in Krebs cycle. So, assertion is glycolysis is the prerequisite for fermentation. Definitely glycolysis is prerequisite for fermentation because pyruvic acid is the substrate for fermentation. End product of glycolysis is the starting material for fermentation. End product of glycolysis is pyruvic acid. It is the starting material for fermentation. Definitely pyruvic acid involves in fermentation. So, both A and R are correct. R is the correct explanation of A. Both A and R are correct. R is the correct. Why? Glycolysis is a prerequisite for fermentation is correct. End product of glycolysis is the starting material for fermentation is correct. What is nothing but pyruvic acid. So, first option. Net gain of ATP in glycolysis per glucose molecule in aerobic respiration is represented as net gain of ATP in glycolysis per glucose molecule in aerobic respiration is represented as. In aerobic respiration in glycolysis 4 ATP are formed but 2 ATP are utilized and 2 NADH2 are formed. This 2 NADH2 is equal to 2 into 2, 4 ATP. 2 into 2, 4 ATP. So, it can be written as in total 4 ATP are formed by substrate level phosphorylation, 4 ATP are formed from NADH2 by ETS. So, in total how many ATP are formed? 8 ATP are formed. In the starting stages 2 ATP are utilized. So, in total the net gain in aerobic respiration of glycolysis is 6 ATP. So, net gain of ATP in glycolysis per glucose molecule in aerobic respiration is represented as 8 minus 2, 6. How 8? 4 plus 4 by substrate level phosphorylation, 2 by NAD involved in electron transport system. So, 8 minus 2, 6 ATP, first option. The only enzyme of Krebs cycle having prosthetic group as cofactor. The only enzyme of Krebs cycle having prosthetic group as cofactor. That is the only enzyme which is not found in mitochondrial matrix but is present in inner mitochondrial membrane. Succinyl thiokinase is not having any cofactor. Isocitric dehydrogenase does not have any cofactor. Alpha ketoglutarate dehydrogenase do not have any cofactor. But succinic dehydrogenase enzyme has a cofactor FAD, flavin adenine dinucleotides, are the only enzyme of Krebs cycle having prosthetic group as cofactor. So, this is the complex 2 in electron transport system, which is nothing but succinic dehydrogenase. Its structure will be like this, having FAD as prosthetic group and 2 iron sulfur proteins. So, FAD is the prosthetic group of succinic dehydrogenase. So, FAD is the prosthetic group of succinic dehydrogenase. So, the only enzyme of Krebs cycle having prosthetic group as cofactor is succinic dehydrogenase. Prosthetic group of complex which is considered as gateway of electron transport. Prosthetic group of complex which is considered as gateway of electron transport. Gateway of electron transport. Gateway of electron transport is nothing but complex 1 or NADH dehydrogenase complex. NADH dehydrogenase complex. In this, the prosthetic group is FMN. So, gateway of electron transport is nothing but complex 1, NADH dehydrogenase complex. In this NADH dehydrogenase complex, the prosthetic group is always FMN. FAD is for succinic dehydrogenase, Cytochrome C is a mobile carrier, NAD is not. So answer flavin mononucleotide, third one. Study the following table with reference to different steps of aerobic respiration and identify the correct combination. Step 1, glucose products ATP, NADH plus pyruvic acid. Definitely in aerobic respiration, step 1 is glycolysis, where glucose converts into NADH2, pyruvic acid and liberates 2 ATP. Step 2, pyruvic acid. Step 2, pyruvic acid, NADH acetyl phosphate. It is wrong. In Krebs cycle, acetyl phosphate is not formed. Acetyl phosphate is formed in phosphoroclastic cleavage of pyruvic acid, which is discussed in biological mechanism of biological nitrogen fixation. Step 3, acetyl coenzyme A results in the formation of FADH2, NADH2, CO2. Definitely this is Krebs cycle. Step 4, NAD, FAD, water and ethyl alcohol is wrong. Step 4 is electron transport system. NAD and FAD is correct, but water is correct, ethyl alcohol is wrong. So A and C are correct. What are A? Step 1 is glycolysis, where glucose converts into ATP, NADPH2 and pyruvic acid. Step 2 is oxidative decarboxylation of pyruvic acid. Pyruvic acid produces NADH2 but not acetyl phosphate. It produces acetyl coenzyme A. Step 3 is Krebs cycle. Acetyl coenzyme involves FADH2, NADH2, carbon dioxide are products, correct. Step 4 is electron transport system. NAD and FAD is correct. Water is correct, but ethyl alcohol is wrong. So here the correct combinations are A and C. The wrong combinations are B and D. So study the following table with reference to depths of aerobic respiration is this one. Match the following with reference to TCA cycle. Product of first oxidation, product of first decarboxylation, substrate of third oxidation, product of second oxidation. Product of first oxidation in Krebs cycle means, first oxidation in Krebs cycle always means isocitric acid converting into oxalosuccinic acid. So isocitric acid converting into oxalosuccinic acid is correct. So definitely A is 5. Then B, product of first decarboxylation. Whenever oxalosuccinic acid undergoes decarboxylation in the presence of oxalosuccinic decarboxylase, definitely alpha ketoglutaric acid is produced. So B is always 1. Then substrate for third oxidation. Substrate for third oxidation means succinic acid converting into fumaric acid is third oxidation, where instead of NADH2, FADH2 is liberated. So definitely succinic acid. And product of second oxidation. Whenever second oxidation takes place, alpha ketoglutaric acid converts into acetyl succinyl coenzyme A, NADH2 is liberated. So A4, B1, A5, B1, C4, D2. A5, product of first oxidation, oxalosuccinic acid. B1, product of first decarboxylation, alpha ketoglutaric acid. Product of or substrate for third oxidation, succinic acid. Product of second oxidation, alpha succinyl coenzyme A. Answer is 1. Higher plants cannot utilize molecular dinitrogen directly. Till now we discussed the bits of respiration. Now we are entering the bits of nitrogen metabolism. Almost all important aspects regarding glycolysis, Krebs cycle, electron transport, oxidative decarboxylation of pyruvic acid, fermentation, RQ. Almost all important aspects of that are covered. Now higher plants cannot utilize molecular nitrogen directly. Why? Because molecular nitrogen, now we are discussing nitrogen metabolism. The molecular nitrogen in nature has very popular triple bond, which is broken only by dinitrogenase. This dinitrogenase is absent in higher plants, but surprisingly it is present in some bacteria and blue-green algae. So that's why bacteria and blue-green algae fix molecular nitrogen. Higher plants cannot absorb molecular nitrogen. Why? Because the higher plants do not have that particular nitrogen called dinitrogen. So higher plants cannot utilize molecular nitrogen directly is correct, because they cannot break the strong triple bond. Higher plants do not have dinitrogenase enzyme is correct. So both A and R are correct. R is the correct explanation of A, first option. Now coming to a matching. Lichens, azolla, stem glands has symbiotic structures. Frankia. Lichens, symbionts with phycobionts and mycobionts. Whenever an algal member and fungal member combines, it is called lichen. The algal member of lichen is phycobiont, the fungal member of lichen is mycobiont. Then azolla. Azolla is a diazotroph in leaves. Azolla contains a diazotroph Anabina azollae in its leaves. Azolla is a free floating fern that help in nitrogen fixation in rice fields, whose importance was first proved in Vietnam. Stem glands has symbiotic structure is Ganira. Stem glands, nostoc is present in the stem glands of Ganira. First option. Frankia, symbiosis with Myrica. Frankia, symbiosis with Myrica, third option. So answer is A5. Lichens, symbionts with phycobionts and mycobionts. B2, azolla, diazotroph in leaves. What is the diazotroph? Anabina azollae helping in nitrogen fixation. And hence azolla pinnata is also resistant to various heavy metals. That's why it is grown near the rice fields, near the factories which are liberating heavy metals. Stem glands has symbiotic structures is Ganira. The gland contains nostoc. Frankia, symbiosis with Myrica. Frankia is an actinomycetes found in the root nodules of non-leguminous plants like ulnas, casuarina, Myrica. So answer A5, B2, C1, D3. Study the following and identify the correct match. Anabina, Frankia, Rhizobium, nostoc. Thallus of Anthoceras, root nodules of Parasponia, root nodules of Casuarina, thallus of lichens, mesophyll cavities of azolla. A4. Anabina is present in the thallus of lichens. It is present in the thallus of lichens. B3. Frankia, root nodules of Casuarina. Just now I told non-legumes like Casuarina, ulnas and Myrica contains a special actinomycetes called Frankia. Rhizobium. 2. Rhizobium is also found in root nodules of a non-leguminous plant called Parasponia. It is also found in the root nodules of a non-leguminous plant called Parasponia. And D1, nostoc. Nostoc is present in the thallus of Anthoceras. Anthoceras is a bryophyte which helps in nitrogen fixation. Anthoceras is also asked as the only algal member with pyrenites or a bryophyte with pyrenites. So fourth option A4, B2, B3, C2, D1. Assertion. The proton and electron source for electron reduction dinitrogen is dinitrogenase. The proton and electron source for the reduction of dinitrogenase is wrong. Source is pyruvic acid, not dinitrogenase. The reduction of dinitrogen to ammonia requires electrons and protons. That is correct. 2 molecules of N2 takes 8 electrons, 8 protons and 16 ATP and produces 2 molecules of ammonia. So for reducing molecular nitrogen to ammonia, 8 plus 8 electrons are required. But the source of these 8 electrons and 8 protons is not dinitrogenase. It is pyruvic acid in majority of the plants, which is nothing but biological nitrogen fixation. So the proton electron source of reduction of nitrogen is dinitrogenase is wrong. The reduction of dinitrogenase to ammonia requires electrons and protons is correct. So A is true, R is false. The phytohormone which induces cell division in the cortex of legume roots is secreted by cortical cells of the root, endodermal cells of the plant, bacterial cells, cells of medulla. The phytohormone auxins are synthesized when infection thread reaches the cortex during the formation of root nodules in legumes. The phytohormone which induces cell division in the cortex of legume roots, which makes the formation of root nodules, is cortical cells of the root only, not by endodermal cells of the plant or by bacterial cells or by cells of medulla. It is only cortical cells of the root. Bacteria only makes the cortical cells to produce phytohormones. Higher plants cannot make use of atmospheric nitrogen directly. higher plants cannot make use of atmospheric nitrogen directly. Just now I told why, because popular dinitrogen has a very strong triple bond which is broken only by nitrogen. In higher plant, dinitrogen enzyme is non-functional. In higher plants, dinitrogen enzyme is absent. How it is non-functional? So A is true, R is false. Third option. A carbon fixing prokaryote which can utilize the first and second abundant gases of the nitrogen. A carbon fixing prokaryote which can utilize the first and second abundant gases. What are first and second abundant gases? CO2 and nitrogen. Both are utilized by only photosynthetic components, and that too a photosynthetic component which can utilize both is nostoc, because nostoc is blue-green algae which can perform photosynthesis, so it utilizes CO2. Nostoc is containing dinitrogenase, so it can utilize nitrogen. Azotobacter is a bacterium which only takes nitrogen. Clostridium only nitrogen. Rhodospirillum only carbon, not nitrogen. So a carbon fixing prokaryote which can utilize the first and second abundant gas of atmosphere, nitrogen and carbon dioxide of atmosphere, or nitrogen and oxygen, is nostoc. Second option. This is a tabular form. Match the following list related to nitrogen cycle. What is nitrogen cycle? Molecular nitrogen is fixed in the soil either by physical methods or by biological methods. This is fixation. How they are fixed? As nitrites and ammonium ions. These are taken by plants. They convert into ammonia. Ammonia convert into amino acids. Amino acids into proteins. Proteins are taken up by animals. So this is nitrogen fixation and this is nitrogen assimilation. Then proteins in animals and plants get converts into ammonia. This is called ammonification. Ammonia gets converts into nitrates. This is called nitrification. Ammonification. Then molecular nitrogen, denitrification. This is called nitrogen cycle. Now, nitrogen assimilation, ammonification, then nitrification, denitrification. 1B, nitrogen assimilation, formation of organic nitrogen. 2C, ammonification is Bacillus mycoides, Bacillus vulgaris and Bacillus rhinosus. Then, this is nitrogen cycle which is very important. Then, nitrification, D, nitrococcus. Nitrification is always done by nitrosomonas which converts ammonia to nitrites. Then nitrites are converted into nitrates by nitrococcus. Denitrification is by micrococcus denitrificants or thiobacillus denitrificants. So answer is 1. 1B, 2C, 3D, 4E. What is nitrogen assimilation? Formation of organic nitrogen. Ammonification is done by Bacillus mycoides. Nitrification is done by nitrococcus. Denitrification is done by micrococcus denitrificans. Match the following: Clostridium, nostoc, Azotobacter, Rhodospirillum. Anaerobic photosynthetic, anaerobic and non-photosynthetic, symbiotic and non-photosynthetic, aerobic and non-photosynthetic. A3, Clostridium is anaerobic and non-photosynthetic. Nostoc, B1, aerobic and photosynthetic. Azotobacter, 5, aerobic and non-photosynthetic. Rhodospirillum, 2, anaerobic and photosynthetic. Identify the incorrect statement regarding dinitrogenase. The synthesis of dinitrogenase is directed by NIF genes is correct. It gets controlled supply of dinitrogen. It gets controlled supply of dinitrogen from leg hemoglobin is wrong. It gets controlled supply of oxygen, not dinitrogen. It accepts electrons and protons directly from pyruvic acid is wrong. It accepts electrons and protons not from pyruvic acid but from ferredoxin. It is very sensitive to oxygen. It is very sensitive to oxygen. It is not incorrect. It is correct. So answer is 2 and 3. Incorrect are it gets control supply of dinitrogenase from leg hemoglobin is wrong. It accepts electrons and protons directly from pyruvic acid is wrong. It does not accept electrons and protons directly from pyruvic acid, but it takes electrons and protons from ferredoxin. So 2 and 3 are incorrect statements. Arrange the following events in nitrogen cycle in correct events. Ammonification, nitrification, nitrogen assimilation, denitrification. Definitely first nitrogen assimilation, then definitely ammonification, then nitrification, then denitrification. So answer is 3 first nitrogen assimilation, 1 next ammonification, 2 next nitrification, and last 4th one denitrification. These are the 4 important steps in nitrogen cycle. The following are related to rhizobium during nodule formation in legumes. During nodule formation in legumes. Flavonoids, phytohormones, curling factor, phytohormones. Flavonoids, cellulases, curling factor, phytohormones. Answer is 2 and 3. Why? Because after reaching the root nodules, they produce a curling factor which converts the root hair into curved structure, which is popularly called shepherd's crook. Then cellulases are synthesized or secreted to make a small pore in the cell wall of root hair. Through this root hair, bacteria enters the root. After entering the root hair, the cell membrane of root hair invaginates around the bacteria to form an infection thread. Infection thread runs up to cortical cells. In this movement, bacteria divides and produce bacterioids. What is the difference between bacteria and bacterioids? Bacteria is rod shaped, present in the soil without functional nitrogenase. Bacterioids are circular, present in the cortical cells, and they have functional nitrogenase. So the following are related to rhizobium during nodule formation in legumes. Flavonoids, cellulases, curling factor, phytohormones. Only rhizobium near the root hair, so cellulases and curling factor 2 and 3. During biological nitrogen fixation, which of the following is used in 1:2 ratio? Which of the following are used in 1:2 ratio? 1:2 ratio, just now we wrote 2N2 plus 8 electrons plus 8 protons plus 16 ATP produces 2 ammonia. So 8 electrons, 8 protons, 16 proteins. So 1:2, electrons 1, protons 1, ATP 2. So electrons, protons, and oxygen will be 1:1:2. So here ATP and O2 is wrong. ATP and O2 is wrong. N2 and H2 is wrong. Electron and proton is not 1:2 ratio, it is 1:1 ratio, 8 protons and 8 electrons. Protons and ATP, 8 protons and 16 ATP. So definitely 8:16 is 1:2. So during the biological nitrogen fixation, which of the following is used in 1:2 ratio means protons and ATP, 4th option. Find out the correct statement. Rhizobium of soil respires anaerobically. Rhizobium in the soil respires anaerobically is wrong. It is an aerobic bacterium. Each codon of genetic code can represent 3 nitrogen bases. It is also wrong. Each codon of genetic code can represent 3 amino acids is wrong. 3 nitrogen base is correct. So each codon codes only 1 amino acid. Dinitrogenase is present in all prokaryotes. It is also wrong. It is present in only few bacteria and blue-green algae. A bacterioid derives its peribacterioid membrane from host cell. Whenever a bacterioid is formed by the replication of bacteria in the infection thread, Circular bacterioids are formed which are surrounded by a membrane which is popularly called peribacterioid membrane, which is obtained not from the bacteria itself but from the host cell. So, rhizobium of soil respires anaerobically is wrong. Each codon of genetic code can represent 3 amino acids is wrong. It is 3 nitrogen bases. Dinitrogenase is present in all prokaryotes is wrong because it is absent in some prokaryotes, present only in few prokaryotes. A bacterioid derives its peribacterioid membrane from the host cell is correct. So, fourth option is correct. In relation to abiological nitrogen fixation, which of these statements are supposed to be correct? It is a physico-chemical process, correct. Physical changes and chemical changes occur one after the other. Nitrogen first oxidized into nitric oxide. Definitely nitrogen is first oxidized into nitric oxide. Then nitric oxide is oxidized into nitrogen dioxide. Abiological nitrogen fixation means physical nitrogen fixation which occurs in 4 steps. First, nitrogen get oxidized in the presence of thunders and lightning to form nitric oxide. Then nitric oxide again get oxidized to nitrogen dioxide. Then nitrogen dioxide combines with rainwater to form nitric and nitrous acids. Then fourth, nitric and nitrous acids reach the soil, combines with their salts to form nitrates and nitrates. So, ultimately what are produced? By biological nitrogen fixation, all the 4 steps involved in the formation of nitrates and nitrates. It is done nowadays in the laboratory also by using a special method called Haber and Bosch process, where 400 degrees centigrade and maximum pressure is used. So, in relation to abiological nitrogen fixation means physical nitrogen fixation, it is a physico-chemical process, correct. Nitrogen first oxidized to nitric oxide, correct. Nitric oxide is oxidized into nitrogen dioxide is correct. Nitrous and nitric acids formed by reacting with calcium or potassium salts to form calcium and potassium nitrates is correct. So, 1, 2, 3, 4 all are correct regarding this one. Now, study the following table and identify the correct combinations. Cycas, gymnosperm, anabina, cycadacearum in the root nodules of coralloid roots are what you call in the coralloid roots is correct. So, Cycas, gymnosperm, anabina, cycadacearum, plant, group and nitrogen fixing organism. Plant is Cycas, group is gymnosperms, nitrogen fixing organism is anabina, cycadacearum or nostoc punctiform is correct. Azolla is a pteridophyte with Anabina azollae. Anabina azollae, because of the presence of Anabina azollae, Azolla pinnata is resistant even to heavy metals like cadmium, chromium, lead, mercury, so many components. Parasponia, an angiosperm rhizobium species. It is a non-leguminous angiosperm where rhizobium lives in the root nodules of Parasponia. Gunnera, angiosperm nostoc. Gunnera is an angiosperm with stem gland showing nostoc. So, Cycas, a gymnosperm with Anabina cycadacearum is correct. Azolla with is a pteridophyte with Anabina azollae is correct. Parasponia is an angiosperm with rhizobium species because it is a non-legume with rhizobium. Gunnera is an angiosperm with nostoc. So, all the 4 are correct. Answer is 1, 2, 3, 4. Bacterioids differ from rhizobium in all except big size, presence of functional dinitrogenase, presence of peribacterioid membrane, being unicellular prokaryote. Both bacterioids and rhizobium are differing in size, differing in the presence of functional nitrogenase, dinitrogenase, differing in the presence of peribacterioid membrane. These 3 are found in bacterioids, not in rhizobium bacteria. But being unicellular prokaryotes is common for both. So, being unicellular prokaryotes is common for both. Fourth option. Central dogma of molecular biology refers to the flow of genetic information from. Central dogma of molecular biology refers to the flow of genetic information from DNA to protein through master copy of genetic information wrong. DNA to protein through working copy of genetic information is correct. Why? Because DNA by transcription in nucleus produces mRNA. mRNA by translation in cytoplasm produces protein. Because of this function, DNA is popularly called central dogma. So, why it is called central dogma? Because DNA to protein through working copy of genetic information. This is the working copy of genetic information. mRNA is called working copy of genetic information. DNA to protein through master copy of genetic information is wrong. DNA to protein through master copy of genetic information is wrong. DNA to protein through interpreter of genetic information is wrong. Working copy to protein through master copy of genetic information is wrong. So, answer is DNA to protein through working copy of genetic information, second option. Reducing power required for the reduction of one molecule of dinitrogen to two molecules of ammonia is 6 electrons, 6 protons and 16 ATP. It is not 6, it is 6 electrons, 8 protons and 16 ATP, but it is converted now into 8 electrons, 8 protons and 16 ATP. In many nitrogen fixing organisms, the reducing powers electron and proton for nitrogen fixation is supplied from in biological nitrogen fixation, a concept is discovered, that is pyruvic acid undergoes a special concept called phosphoroclastic cleavage. Phosphoroclastic cleavage, by which 8 electrons and 8 protons are liberated, and pyruvic acid forms acetyl phosphate and CO2. These 8 electrons are taken by ferredoxin. Ferredoxin by alternate reduction and oxidation gives electrons to ferredoxin. From ferredoxin, iron, electron goes to molybdenum iron protein. Finally, the electrons are given to 2 nitrogen. In the form of a cyclic manner, they go 2 nitrogen and forms 2 molecules of ammonia. So, 8 electrons move like this. But protons are directly added here. This is biological nitrogen fixation, where dinitrogenase is a special component with 2 parts. One is a dimer, that is iron protein part. The other is a tetramer, that is molybdenum iron protein part. So, in this type, what you call, in many nitrogen fixing organisms, the reducing power electron and proton of nitrogen fixation is ultimately supplied from, not from leg hemoglobin. Leg hemoglobin is a pink colored pigment present in the root nodules of legumes, where whenever there is an oxygen, dinitrogenase cannot show its function. That oxygen must be removed from dinitrogenase, but that oxygen must be supplied to rhizobium for its survival. So, this careful, very sensitive situation of supplying oxygen to rhizobium and making the nitrogenase present in rhizobium not to get exposed to oxygen, leg hemoglobin functions like a biological curtain. A biological curtain. Leg hemoglobin functions like a biological curtain. And leg hemoglobin also popularly called oxygen scavenger. And this method of supplying oxygen carefully to rhizobium and making the oxygen not to get close to the nitrogenase enzyme is popularly called biological compartmentation. This is also called biological compartmentation. So, leg hemoglobin is biological curtain or oxygen scavenger or helping in biological compartmentation. Dinitrogenase is an enzyme which helps in nitrogen fixation. Ferredoxin is the component where they takes electrons from pyruvic acid and then by alternate oxidation reduction through iron protein and molybdenum iron protein, it gives to molecular nitrogen and reduce it into ammonia. So, in many nitrogen fixing organisms, the reducing power electrons and protons for nitrogen fixation is ultimately supplied from pyruvic acid. In the mechanism of nitrogen fixation, reduced ferredoxin donates its electrons to. It is not donating electrons to nitrogen, it is not donating electrons to molybdenum iron tetramer, but not to acetyl phosphate. It gives electrons directly to an iron part, that is dimer. So always nitrogenase enzyme has two parts. One a dimer part called iron protein part, a tetramer called molybdenum iron protein part. So in mechanism of nitrogen fixation, reduced ferredoxin donates the electrons always to iron dimer. Arrange the following activity, following events in nitrogen metabolism in a sequence. Synthesis of amino acids, formation of nitrate from nitrate, formation of ammonia from nitrite, formation of polypeptide. First, formation of nitrite from nitrate. First, formation of nitrate from nitrate. Then formation of ammonia from nitrite. Formation of ammonia from nitrate. Then ammonia is converted into amino acids. Then amino acids are linked by peptide bond, resulting in the formation of a polypeptide chain of amino acids called proteins. So first, formation of nitrite from nitrate. Reverse direction, formation of nitrite from nitrate. Formation of ammonia from nitrite. Formation of ammonia from nitrate. Synthesis of amino acids. Ammonia is converted into amino acids. And formation of polypeptides. So 2, 3, 1, 4. After the formation of amino acids only polypeptide link takes place, and a long polypeptide chain of amino acids is called as protein. Arrange the following events in sequence during root nodule formation. Shepherd's crook, flavonoid secretion, infection thread formation, cellulose secretion. First, flavonoids are secreted. Whenever flavonoids are secreted, rhizobium goes there. Whenever rhizobium goes there, Shepherd's crook formation occurs. Whenever Shepherd's crook formation occurs, cellulose formation or cellulose liberation takes place. Once the cellulose liberation takes place, small hole is made. Small hole is made, and finally it was. So first is 2, flavonoid secretion. Next is 1, Shepherd's crook. Then is 4, cellulose secretion. Finally is infection thread formation. So answer is 4. 2, 1, 4, 3. How? First, flavonoid secretion. Amino acids, flavonoids and carbohydrates are secreted by root of legumes, which are liked by rhizobium, which come close to the root. After coming to the root, root identifies whether it is not rhizobium or rhizobium by secreting special components called lectins. Then root hair number increases by rhizobium. Now rhizobium produces curling factor, which makes the root hair to curl at the tip, which is called Shepherd's crook. Now cellulose formation occurs, cell wall made a small pore, rhizobium enters and a plasma membrane invaginates around rhizobium, resulting in the formation of infection thread. Once the infection thread is formed, it runs up to cortex, and in the cortex bacterioids are liberated and root nodule formation occurs. Then vascular connection is formed between root nodule and root system, vascular system, and this is root nodule formation. So answer is flavonoid secretion, Shepherd's crook, that is not Cook's, that is Shepherd's crook, cellulose secretion and infection thread formation. Fourth option. Host, microorganism, symbiotic structure. Cajanus, rhizobium, root nodules. Cajan belongs to legumes. Definitely in legumes rhizobium will be there, and definitely this rhizobium will be there in root nodules. Cajan, red gram. Microorganism present in this is rhizobium, and where it is present, definitely root nodules. Casuarina, frankia, root nodules. In Casuarina, frankia is not present. In Casuarina, frankia is present, but symbiotic structures, root nodules. Now, Cycas, azospirillum, coraloid root is wrong. Cycas, azospirillum, coraloid root is wrong. Ganira, anabina, stem glands is wrong. Ganira, anabina, not anabina, it is nostoc. So, Cajan, rhizobium, root nodules is correct. Casuarina, frankia, root nodules is correct, because frankia is an actinomycetes membrane present in the root nodules of Casuarina, ulnus and myrica. Cycas, azospirillum is wrong. Cycas is Anabina cycadacearum or nostoc punctiformis. Correlate root is correct but not azospirillum. It is either Anabina cycadacearum and Nostoc punctiformis. Ganira, Anabina, stem glands is wrong. Stem glands is correct. In the place of Anabina, it must be Nostoc. So, these are some of the bits in nitrogen metabolism, some of the bits in respiration. Almost all important aspects in respiration and nitrogen metabolism are touched, and these bits are very important, pulled up from the syllabus and given and studied and also discussed in the class. In the next class, we discuss the remaining nitrogen metabolism, that is starting from genetic code to protein synthesis and if possible phytohormones. So, next class it will be genetic code, protein synthesis, phytohormones, and then we discuss biotechnology, genetic engineering, phytohormones and if possible crop improvement also. And next biotechnology, genetic engineering, finally mushroom cultivation, single cell protein and all. Okay.